name is Suzy Buckley. I'm a lecturer in neonatal gene transfer here at the Institute for Women's Health at UCL. I've been working in the field of neonatal gene therapy for over 15 years now, and I've got a special interest in targeting treatments to the lungs to treat pulmonary disorders. So this includes things like CF, cystic fibrosis, which was one of the first diseases to be put forward for gene therapy treatment, and also things like surfactant deficiency and bronchopulmonary dysplasia. Most of my research is carried out on mice. They make really good preclinical models for neonatal therapies because they have short gestation periods and large litters. Also, there are lots of transgenic mouse models available of these genetic diseases, so we can test the different gene therapy treatments on them. The basic principle of gene therapy is to replace the mutated gene sequence that is causing the disease with a healthy copy of the gene containing the correct DNA sequence. To do this, we use genetically modified viruses, things like lentiviruses or adeno-associated viruses, to deliver the gene to the cells where it's needed. And there are several advantages to treating these diseases in the neonate rather than in the adult. So with early onset diseases, you can aim to provide treatment before any pathology has occurred. Neonates have a less mature immune system, so you can see reduced problems of immune rejection of corrected cells than you do in the adult. And quite simply, neonates are much smaller than adults, so you require less viral vector. I'm also using these neonatal gene delivery techniques to create and study disease processes in living animals in a technique that we've called somatotransgenic bioimaging. So here we use the same gene therapy viruses to deliver the luciferase marker gene to a particular organ or tissues within the neonatal mouse. And the expression of this luciferase gene is under control of a promoter that's involved in a specific signalling process or disease pathway, so something like NF-kappa B or TGF-beta. So when you stimulate these mice or induce the onset of disease in these animals, the promoter is activated, the luciferase gets expressed and light is produced. And you can simply measure this light using a fancy expensive camera which allows you to monitor these signalling changes in living animals in real time. So this new technique allows you to learn even more about the underlying mechanisms of the disease and also even to test different therapeutic compounds. You can read more about biosensing and the rest of my research in my publications, and if you're interested and would like to know more or have any questions, then please get in touch.